Hi, everyone. Welcome back. Today, I have for you a video that's potpourri, a hodgepodge of different topics that we just haven't finished over the past two lessons. But I figured the best place, the most logical place to start would be with discussion of our continuing our discussion from Monday, trying to finish up that lesson on protein structure. So if you recall, we had talked a lot about the amino acids that make up the primary structure. And then we started discussing how those individual amino acids, when they're linked by peptide bonds, can take on very specific dihedral angles that allow the formation of alpha helices and beta strands. And remember, the benefit to this is that when you form those alpha helices and beta strands, the peptide backbone is able to form all these different hydrogen bonds. And when you have hundreds and hundreds of these hydrogen bonds, that's what stabilizes a protein structure, among other things, but definitely the sum of all those hydrogen bonds is important. Okay, and that's why we see these structures over and over and over again in protein structures. You see alpha helices and beta strands pretty ubiquitously. Okay, very few residues within a protein will be what we call disordered or not participating in one of these structures here. Okay, so that brings us up to secondary structures, and I'd just like to go into a little bit more detail on each of these, give you some guidelines on what a good alpha helix looks like, a good beta strand, for example. So let's start with the alpha helices. Just some interesting geometric facts for you. An alpha helix has about 100 degrees of turn per amino acid. So that means that we'll need about 3.5 amino acids to complete one rotation of that helix. And that helix, by the way, is very solid. It takes up, the atoms within the helix take up a lot of its volume, and that's what you can see over here. Okay, so we have very little free space, which is good because then that can also open the door for things like van der Waals interactions. That's where you get slight dipoles even between carbon atoms that help to provide some weak attractive forces. Okay, so I wouldn't want to have large empty spaces inside of my protein or inside of my helix. It just wouldn't make sense. That would be a missed opportunity to have new, even if they're weak, but new types of bonds that are holding everything together. Okay, there is a little bit of a cavity here, but it's not very big. But that being said, the helix is still stretchy. So it does still allow the protein to move under different circumstances, to adopt different conformations, okay? And the length of an alpha helix can be quite long, about 40 amino acids long, I believe is the largest reported alpha helix, but they tend to be much shorter than that, including about 10 to 20 amino acids, okay? But what's really interesting too about these structures is that whenever you form the helix with the backbone, the amino acid residues, the aspartates, the glutamates, the serines, the etc., right? They all point outward from the helix. Nothing points inward here. Everything is pointing out away from the helical scaffold. And not only that, the residues also have an angle to them. So they're not pointing straight out from the helix, they're pointing slightly down or towards the N-terminus would be the better reference here, okay? So you can liken this to a Christmas tree where the C-terminus would be the top, the N-terminus would be down here at the bottom, and you know the branches of a pine tree or Christmas tree, they stick out, but they definitely hang low from gravity, and the residues do the same thing here. Okay, so that's what you can expect to see is these residues pointing out and down from that helix. Okay, just to give you an idea of what these things actually look like. And last but not least, a lot of times when we need to describe a helix or the residues within it, we'll use this type of top-down diagram here. where you can see the N-terminus is on the outside of this spiral. And then the spiral continues onward. And what you can see here is, for example, here's the first residue of the helix. That N would be an asparagine. Here's an isoleucine, that's the second. Alanine is the third. Glutamate is the fourth. Okay, so on and so forth. What this diagram is actually showing you is if you're looking at the top of the helix down, where those residues would be sticking off relative to one another. Okay. Now, initially, you might wonder, all right, Dr. Nomer, that looks like a neat diagram, but why in the world do I need to know that? Well, what you'll find is that a lot of alpha helices, they're actually what we call amphiphilic, which means that they have at least one face that is hydrophobic and another face that is hydrophilic. Because you might have a protein here, it's a large protein, okay, and have an alpha helix right here, where one side is oriented towards the solvent and needs to be polar or charged, whereas the other face is going to be contacting the rest of the protein, and that needs to be hydrophobic. And so in this diagram here, the polar or charged residues are labeled in light blue, hydrophobes are labeled in dark red, and you can see what I'm talking about, right? If I'm looking at this helix from the top down, half of it is pretty hydrophobic. That's the hydrophobic face that would be oriented towards the core of the protein. The hydrophilic face here would be oriented towards the solvent, okay? So it's a nice, quick, easy way to describe, what residues are in the helix and what their character is. Okay, so that's the geometry of a helix and of an alpha helix. Now let's talk about the constituent amino acids. So like I was just saying, you'll typically find in most proteins that half of them will be hydrophobic, half of them will be hydrophilic. But that's not always the case, for sure. There are plenty of proteins, for example, that have alpha helices that are embedded in the cell membrane. Those residues are almost exclusively hydrophobic because the cell membrane is a hydrophobic domain. But anyways, more generally speaking, there are some residues, whether they be hydrophilic or hydrophobic, that we like to see in an alpha helix that provide a stabilizing type of effect, or at the very least, don't destabilize the alpha helix, okay? So for example, we like to see hydrophilic residues that are polar and flexible, arginine, lysine, glutamate, and glutamine, okay? Same thing with hydrophobics. We like to see some flexibility there too, but smaller ones are also tolerated. Okay, so things like methionine or leucine. Okay, the reason we like flexibility here is because if you look at an alpha helix, a lot of these atoms in the backbone are going to be participating in hydrogen bonds, but the more hydrogen bonds, the merrier, right? And so if we have a long polar molecule here, let's say it's even negatively charged, okay, or it's got an oxygen that can participate in hydrogen bonds, if it's flexible enough, there's going to be some disordered residues here at the end of that alpha helix, and this residue can bend over and participate in some hydrogen bonding there, okay? So that's what I mean when I say flexible residues are preferred. That's because those longer residues can interact with more atoms, and that gives you the possibility to create additional bonds, okay? But even so, most of these are just very weak rules of thumb for things that are good to have in an alpha helix. What I'd like to point out, more importantly, are things that are bad to have, things that destabilize an alpha helix, okay? So for example, I would not want to see a proline, a glycine, or any of these beta-branched amino acids, valine, threonine, or isoleucine, in an alpha helix. And the rationale for that is multiple fold. First of all, proline. Remember, proline is this oddball amino acid that puts a kink in the backbone. Remember, we had to have very specific dihedral angles to achieve that helical structure. You can't achieve those with a proline. So proline will pretty much prevent that spiral wrapping of the alpha helix. So we tend not to see prolines within an alpha helix. Now, that's not to say it's impossible. You will definitely find alpha helices in nature with a proline smack dab in the middle of them. But if you have the chance, if you're choosing to mutate that protein, it would probably be a good idea to get rid of that proline. You might significantly increase its stability. Okay. Now, glycine, on the other hand, is the more flexible residue, right? So why would we say that's a bad thing to have in a helix that is known to be stretchy? Well, the issue, again, just like we said before, is that glycines don't really contribute anything to the protein structure. And there's going to be a lot of other residues exposed in this alpha helix. It would be nice if we had something there instead of a glycine, another residue that could introduce new bonds. So you'll see that time and time again. We really try not to use a glycine unless we are in a region that needs to be flexible. If you put glycine anywhere else, you're missing something that could be good, right? The opportunity to make new bonds. Then over here, we have the beta branches. Remember, the idea here is that if you look at the backbone, the first carbon is the alpha carbon on the side chain. The second one is the beta carbon. And we have some of these residues like valine, threonine, and isoleucine that have two carbons coming off of that beta carbon, the gamma 1 and the gamma 2. A lot of other residues don't have this second carbon, and so they're a lot more flexible. They have a lot less steric bulk, but these guys have the heightened steric bulk, and that is actually something that can interfere with the formation of the helix if you have too much of that bulk close to the peptide backbone. Okay, so just like proline, we tend not to see valine, threonine, and isoleucine in these alpha helices. Again, you'll probably go into nature and find an alpha helix that has one or more of those residues, but if you can engineer a protein to remove them, then your alpha helix will probably be more stable. Okay, so take everything on this slide with a grain of salt. You can optimize a protein by making these changes, following these guidelines, but they're not absolutely mandatory. Okay, so there you go. I don't want to see proline, glycine, or beta branched residues in an alpha helix. Anything else is probably fair game. Anything that creates new bonds is definitely preferred. So that's an alpha helix, but most proteins usually have multiple alpha helices, right? Typical protein is hundreds of amino acids long. Most alpha helices are 10 to 20 amino acids long, so there must be several of them, right? That contribute to a ternary structure for a protein. So if that's the case, then how do we connect those different alpha helices? Well, we don't have to be too sophisticated about it. As it turns out, the domains that connect these alpha helices are called loops, and they're fairly disordered. They don't have a lot of hydrogen bonds formed in their peptide backbone. So they're very loose. They're very flexible, which is exactly what you need if you're going to make an alpha helix, for example, that's pointing off in one direction, but you have to get that back to the protein to make the next alpha helix, right? You want to have some flexibility here so you can connect those two helices and keep them together in the overall protein. So that being said, the connectors, the loops between these helices are usually long and flexible. Could be about 2 to 10 amino acids. That's what I mean by long. We'll talk about the domains that connect beta strands later. Those are called turns, and they're usually only 2 to 3 amino acids long. So that's why this is relatively long, these loops, okay? And the reason I talk about them now is because in these loops, they're very tolerant of mutations. You can put pretty much anything you want in these loops, and since they weren't already contributing, for instance, to an alpha helix or a beta strand, you don't have the same limiting rules, right? You could put a proline there, and it's not going to disrupt a specific structure. So if you're going to mutate a protein and it has a lot of these loops, that might be a good place to start. If you mutate something in an alpha helix, you might have the best intentions in mind. You might have a fantastic model that supports that idea, but you always run the risk of destabilizing that helix and destabilizing the protein overall, okay? So case in point here. Here's an enzyme, cytochrome B562. And it has this loop right here connecting its alpha helices. And the native sequence there is a phenylalanine at position 61, a few more residues, and then a phenylalanine at position 65. All right. And the native protein here binds a heme cofactor. All right. So it's got this exogenous little molecule here that comes in and binds, and that's how we get cytochrome. If you take that loop between the alpha helices and you mutate them, or specifically if you randomly mutate those phenylalanines, one study showed that 91% of the mutants were still functional. That's really unusual. If you were to target an alpha helix or a beta strand, a lot more would be unfunctional, dysfunctional, because they couldn't fold properly anymore. Now, some of the mutants did have altered redox activity, so it did affect the activity of this heme right here. That's what we're saying when we say redox activity. But one mutant, a phenylalanine 65-divaline, actually had a higher activity than the wild type. So there's a lot of opportunity here to improve these structures or improve enzyme activity within these loop structures. Okay, so these loops are malleable, right? We can change them pretty freely, but you might also be wondering, what if I'm trying to optimize the protein structure? What if I want to make it as compact as possible? Well, then you might be interested in designing a minimal loop, okay? So let's say you're making a synthetic protein even, right? You know you have to have these alpha helical structures close together, but what should come between them? Well, we can certainly use very long loops to connect them, but those might be less stable because that's a large disordered part of your protein. So it might make it more prone to aggregation and things like that. So instead, we tend to make loops shorter if we can, okay? And there have been several studies on this. I just want to summarize them real quick here. But what you find there is that glycine mutants fold pretty easily, right? Because those residues are pretty flexible, but the proteins containing them tend to be less stable because you're not introducing hydrogen bonds or ionic bonds that you would get from other types of residues, okay? So yeah, glycine is flexible, but be careful with how you use it. Now, instead, what you want to do is you still want to have that flexibility from glycine, but you want to have something besides glycine in that loop. So for example, these loops down here, like this glycine, threonine, serine, glycine, and an asparagine combination. The threonine and the serine have hydroxyls that can make hydrogen bonds. The asparagine has the same thing. It's got an amine group on there, and it's got a hydroxyl, or no, it's a double bonded O. That's what it is. It's a ketone. Okay. So all of those things can form hydrogen bonds, and that's what makes GTSGN a better loop than GGGGG. Okay. I'm trying to drive this thought home to you, right? That glycine is nice and flexible, but if you can put some things in that contribute new bonds, that's preferable. Okay. And here's just several other examples of that basic idea. All of those loops should be good enough for putting into your protein, okay? But all of them are pretty short. They're still about 2 to 6 amino acids long, okay? So that's what a good loop looks like. So now you know how to design an alpha helix, you know how to design a loop, or at least you know what to look out for, right? If you're trying to optimize the structure of a protein, you can follow these guidelines here. Now let's talk about the other secondary sequence, our structure, beta sheets. All right, so beta strands here, what we want to see here are hydrophobic residues that are very bulky, okay? So all of these residues here, tyrosine, phenylalanine, tryptophan, those are those hydrophobic residues that had large aromatic groups to them. Okay, and the reason why those are favored in beta strands is because, in contrast to alpha helices, where steric bulk was actually disfavored, in a beta sheet, we have just that. We have a sheet that looks something like this. And what I want to do on this sheet is I want to have residues here that can interact with one another, that can form different types of bonds, for example, form hydrophobic interactions with one another. And so if I just had a tiny little valine at each one of these spots, you can see those atoms wouldn't really interact between those residues. No, instead, what would be better is if instead of valine, I had something longer, right? Like here's a big tryptophan there, maybe or no, so that would have been a phenylalanine. Let's put another tryptophan here. Forgive my chemical structures, right? They're pretty rough, but you get the idea. You can see how those large rings really fill up this space. So now I can have a lot of interactions between those residues because a beta strand is really, really spread out, right? It's really just a plane. It's a corrugated plane, but you get the idea. And those residues are, oops, there we go. Let's just draw it like this. Those residues are pretty spread out initially, so they need to be long to contact each other, okay? At the same time, solo hydrophilics can also be bad. Like let's say on the other side of this sheet, I'm gonna have some more residues, R1, R2, for example. If I just had an aspartate, okay, with a negative charge, and then maybe a serine over here, and a serine over here, there's nothing for that negative charge to bind to. And so again, I'm missing an opportunity. What I'd like to have is instead of that serine, maybe I put an arginine right here. So that gives me a positive charge. And now I can form an ionic bond that keeps these things together, okay? It adds some stability to the overall protein. So you'd like to see pairs of ionic residues. You'd like to see flexible hydrophilics that can make hydrogen bonds as well, okay? So we want to pair everything, glutamate with a lysine, aspartate with an arginine, and etc. Proline and glycine are usually found in beta strands, but typically towards the end of each strand. Because once again, proline has a kink in it, right? That can disorder or destabilize the beta strand itself. And glycine, just like always, yes, it's flexible. You could put it in a beta strand, but you wouldn't be getting these bonds that we're getting from the other residues, okay? Oops, yep, there we go. That's everything I have for that slide there. But you get the idea, all right? We're trying to fill space with these beta strands. And before we move on, I'd like to point out that the isoleucine and the valine that we didn't want to have on the alpha helices are actually good to have here on the beta strand because they fill up that space close to the peptide backbone. I would prefer, yes, to have the larger hydrophobes instead of that valine, right? That's the example I gave earlier, but it's okay to have beta branched residues here on your beta strand, whereas it's prohibitive on the alpha helices. Okay, so we had loops to connect the alpha helices, and for the beta strands, the connectors here are turns. Like I said before, turns are very short. They tend to be only 1 to 5 amino acids long, and they also tend to be very rigid as well. Because if I have a beta strand that's going in one direction, and I want to mate it to a beta strand that's going the other way to make a beta sheet, I have to make a very tight turn, a 180-degree turn, in as little as 2 amino acids, okay? So we've got 2 90-degree turns there. And one of the best ways to do that is with a proline. So remember that kink that proline puts into the backbone? That's bad for an alpha helix, bad for a beta strand, but it's very good to have in this situation right here. When I want to have a connector between these two beta strands that's nice and short, proline makes that turn faster than the other residues do, okay? But at that same time, since it's a proline, it's also very rigid. And so that's why I say here that turns tend to be rigid, okay? And they're also highly conserved as well. While I told you to mutate loops, I would not suggest mutating a turn between two beta strands. If you look at, for example, the same enzyme across multiple species, you'll find that the turns, the sequences in those turns really don't change very much. Loops change a lot between species, if you're looking at the same enzyme, that is, but not turns. Okay, so here's an example. Just like I gave you the cytochrome example for the loops, now let's look at plasticyanin here. All right, so this is an enzyme that binds copper. So it's normally got a bright blue color to it. So that's convenient because what they did in this study was they took one of these turns in the enzyme, that's this one right here, okay, that's those residues, and they mutated these residues. So there you go, you can see there's a proline in this turn, and then there's those three other residues as well. A glycine here contributes flexibility, so if it's not a rigid turn, at least it can make that kind of rapid turn with the glycine. Okay, so anyways, that's the sequence that we're going to mutate. We're going to make a bunch of mutations in each of these residues. And the hypothesis is if that mutation is inconsequential, the protein will still fold and it will still have that blue color from the bound copper. But if the mutation is disastrous, if you put some other residue at the position of this proline, for example, then maybe the protein doesn't fold, then it will not have that blue color. It won't be able to bind that copper anymore because the copper binding site, which I think is right there, will be disordered now, okay? So anyways, long story short, if we mutate those residues, out of about 100 of these things, only 6 were still blue, okay? So only about 6% still bound copper. Only 6% were still properly folded. Now, that's not to say they weren't, the other ones weren't all unfolded. No, there were actually 82 that still folded, but were unable to bind the copper. So the protein was still soluble, it just couldn't bind to that Cu, okay? So 88% approximately of these things were dysfunctional. That goes to show you how very important the sequence of these turns are, okay? 10 didn't express at all. Oh, so yeah, I should say not 88%, but about 92% of these mutants were not functional, didn't fold properly, okay? So there you go. Do whatever you want with a loop. Protect your turns. If you're designing a new turn, it should be very short. It should probably have proline, maybe some glycine. And if you put anything else there, it should also be small, like a serine or a valine, or it should be a residue that gives you some new bonds, a hydrogen bond or otherwise, okay? So there you go. Alpha helices, beta strands, loops, and turns. Those are your 4 biggest types of secondary structure, or most common, I should say. All right, so then let's just wrap things up here by talking about the next 2 levels of structure, which are vastly more simple, okay? Now we have tertiary structure. And tertiary structure, that's just the collection of all the different secondary structures. So for example, your chromoproteins and GFP are what we call a beta barrel, which is just another name for a beta sheet that's been wrapped into a cylinder. So here we've got a collection of these beta strands that all come together to form that cylindrical structure. Okay, that's an example of tertiary structure. At the same time, I could show you hemoglobin, which is just a collection of alpha helices. And I think you will see that on the next slide, actually. That's another example of tertiary structure. Yeah, here we go. There's hemoglobin. So it's just a collection of all these alpha helices that assemble into these subunits. Okay. But what you're looking at would be 4 different subunits. So the purple, the red, the blue, and the green, those are independent peptides that all have their own tertiary structure. But then when the hemoglobin subunits assemble into a tetramer, that's now our quaternary structure. Okay. And then also hemoglobin binds heme, just like cytochrome. So you could have a single subunit protein, for example. If it picks up a cofactor like heme or copper or something else, that's another example of quaternary structure. Okay. Now, when we get to this point and we're talking about quaternary structure, it's important to point out that there are several things that could motivate those subunits, for example, to bind to each other. Okay. One of the most common interactions is, let me just get all my animations on here, is something called hydrophobic interactions. Okay. Let's say I had one subunit A and another subunit B. And A is covered in charges and different polar residues, mostly, but then A's got a hydrophobic region here. A bunch of tryptophans, valines, isoleucines, things like that, a bunch of hydrocarbons, not very many nitrogens and oxygens to be seen. B has the same phenomena. It's also got a hydrophobic patch here where there's nothing but hydrophobes, okay? If you put these two things in solution, those hydrophobic patches, what will have to happen there is that water will form a cage around them. Now, this might be a new concept for you, but we often say that hydrophobes don't like to be dissolved in water. So you might assume that water just doesn't interact with them at all, like water would bind to a polar residue like serine, which has a hydroxyl or asparagine or aspartate that's charged, right? The interactions between water and those residues are fairly obvious, right? Because there's going to be charge-based, polar-based operation or interactions. But even so, with hydrophobic residues, water does not just ignore those residues. Instead, it has to form an ordered cage around them. So what you have here is pretty much a phenomenon where water molecules get trapped around hydrophobic residues. Okay, so if these two subunits in this example up here are in water, there's going to be some water molecules that are occupied in those hydrophobic patches. And if we're thinking about the system here in terms of entropy, and by the way, we'll describe a lot more of this in detail next week when we have our lesson on thermodynamics and protein folding. But if we're thinking about entropy, that's a bad thing for entropy, right? Because if those water molecules are constrained to a specific position, a specific microstate, then that means that the entropy of the system with that protein in it will be lower, okay? However, if A and B here, if they interact, if they bind to each other, so now those hydrophobic patches are bound here, they're concealed. What will happen there is that will mean that these water molecules that were caging those hydrophobic patches, now they're released. And that's exactly what you see in this transition in the figure here. There's 6 of these blue dots. These are all water molecules that would be released. And if they're released, that means they can occupy a higher number of microstates. That means the entropy of the system actually increases when those two subunits come together, okay? That's a phenomenon called hydrophobic interaction. So a lot of times it's entropy that motivates the assembly of these protein subunits. You might think it's enthalpy. Enthalpy refers to the formation of bonds, right? Ionic bonds, hydrogen bonds even. But in fact, even the hydrophobic residues can contribute to protein stability. They can contribute to this assembly here by binding to each other and releasing water molecules. All right, so that process is called hydrophobic interaction, and it increases the entropy of the system. Okay, so hemoglobin's a great example of that. There's a lot of hydrophobic interactions at these subunit interfaces. So that's the interaction that's difficult, maybe counterintuitive. are at least not intuitive to understand. The other ones are fairly simple, right? So some other ways to bring these subunits together would obviously be with ionic bonds. Okay, so this is where we get back to something like a lock and key mechanism. The hydrophobic interactions aren't going to be very specific, right? But if I had, let's look at protein A and B again. If I had an exact sequence of charged residues on A like this, and it was negative, positive, negative, but then on B, it was positive, negative, positive, you can see how those charges line up. So A would specifically bind to B here. Okay, as opposed to if I had C and the order of charges on C was all negative, then C probably wouldn't bind to either A or B. Okay, so while hydrophobic interactions are definitely important, ionic bonds can lend specificity to these assemblies as well. Okay, by lining up the different opposing charges, then we can get very specific interactions between one subunit and another. All right. And before we move on, I just want to mention that ionic bonds like these are typically referred to as salt bridges. And within these salt bridges, certainly we have some ionic interactions there, but you might also have some hydrogen bonding that occurs as well. So that's what's very special about lysine and glutamate and all the other charged residues, is that they can not only contribute ionic bonds, but also hydrogen bonds as well. And those interactions collectively are called a salt bridge. Okay. And then last, but certainly not least, we've also got our disulfide bonds. So if you had, again, protein A and protein B, and they each had a thiol on them, you could oxidize those thiols to make a bond here, okay? And that would be very nice to have, because now that you've assembled the protein, you're not going to disassemble that, right? Now there's a covalent bond linking the two peptides. So that's a pretty common phenomenon there in nature. But certainly you see disulfide bonds not only between different subunits, but also within a single subunit as well. There might be buried cysteines, for example, here that add covalent bonds to B and increase its stability. Okay, so there you go. Those are the levels of protein structure, and this is how you keep those structures together, is with one or more of these interactions at every level. Secondary structures are mostly hydrogen bonds, but when we get up to tertiary and quaternary structures, that's when we start to see more advanced or more complex mixtures of these different bonds. Okay, so I had this slide in here just to give you a brief intro to what we're going to talk about next week, but pretty much the purpose of this slide was to say, how do we model all this, right? How do we quantify the stability of a protein? How can we look at a structure and determine, well, this protein structure is more stable than the next one, or less stable, or something like that? Or will this protein fold at a specific temperature, or will it unfold, denature, and aggregate? Well, the answer to all of those questions is pretty much this equation right here. We'll talk about this at length next week, but I just wanted to put it in front of you because I wanted to say that protein folding can be described by the Gibbs free energy of the system, okay? And if the delta G here, which is the combination of the enthalpy from the formation of any bonds upon protein folding and the entropy during that folding process, if that delta G is negative, then the protein will fold. If the delta G is positive, then the protein will not fold, or it will unfold, okay? And what we tend to see here is that the delta H is inherently a negative property. Okay, so that's good. The formation of more bonds will make this delta H more and more negative. But we also see that the delta S is negative as well. Okay, and notice this negative sign in between them. So that means entropy actually disfavors protein folding. That shouldn't be a surprise, right? When a protein folds, each atom is constrained to some specific microstates. So protein folding would actually lower the entropy of a system. And so that's what you see here. The delta S is going to be negative. minus a negative delta S is going to be positive. So delta S is going to be working to make delta G more and more positive, whereas delta H is making it more and more negative. Okay. And notice here too, that temperature affects this relationship. So the higher the temperature, the larger the contribution of delta S to this calculation. And so that can definitely work against you. And you've seen this every time that you boiled an egg, for example, right? You boil the egg or fry an egg, I guess is a better example. Initially, it starts off clear, all the proteins are dissolved, but as you increase the temperature, they reach a point at which delta G is positive instead of negative for them, and they start to denature and aggregate, and the egg turns white, okay? But yeah, so there you go. Brief overview of what we're going to talk about next week. We're going to take much more time to unpack that at that point, but it's delta G that describes if a protein will fold or not. And that, ladies and gentlemen, is how this program right here, Foldit, works. It takes a structure and it can actually calculate the delta G for that system, and it gives you a score. And then all you have to do to make the protein more stable is to maximize that score. Now here they have the score listed as positive, so it's not really a reflection of delta G or a true interpretation of delta G, but it would be confusing if it was negative, right? People will say, hey, why isn't it positive? So yeah, that's what Foldit does. It looks at all those elements of structure that we talked about earlier. Are there hydrogen bonds, disulfides, hydrophobic interactions, for example, or are there repulsions? Are there steric hindrances? It looks at all of that within a protein structure, something like this, and then it gives you a nice arcade-like high score, okay? And the higher that score is, the more stable the protein is. So that's how this group here at University of Washington pretty much gamified protein structural simulations, and it's pretty fun. So that's why I have it assigned to you guys as an assignment here this semester in the course, okay? So what I'd like you to do is go to Foldit, make an account, and go through all the tutorial puzzles, okay? Now, if you need any help with that, I've posted a walkthrough that gives you some pretty helpful hints for getting through some of the tougher tutorials. And I'm also here too, if you need any help. Like if something's not working, let me know. If you tried it for, an hour, that definitely shouldn't be the case. Most of these tutorials should only take 5 to 10 minutes max, okay? The best piece of advice I can give you is if you've been working on a puzzle for more than 5 minutes, think about starting over. Sometimes the protein gets trapped in this conformation where it's never gonna be more stable unless you start from scratch. So yeah, if you're stuck, consider starting over, okay? And it should work out for you pretty easily after that. But anyways, I hope you have fun with this. I hope you enjoy it. And it really helps you to build this intuition for protein structure that we started nurturing on Monday with those 3D printed models. Here, you get to play a video game with it. But I want to leave you with this thought that it's not just a game. Foldit is actually a serious piece of simulation software. Indeed, there was this protease here from the Mason-Pfizer monkey virus that researchers for the longest time knew the primary sequence of that protein, but they couldn't find the structure. And yeah, we'll talk for more, we'll explain reasons why that might be, but the experiments you run to determine protein structure can be very difficult sometimes and almost impossible in some cases. So that's the situation here with this protease. They couldn't find the 3D structure of the protein. So they put it onto Foldit here and just released it to the internet. They crowdsourced the research, the simulation here. And within 10 days, users were able to find the structure shown on the slide. And later, the researchers were able to confirm that this is the structure of that protease, an internet game. did more for finding this protein structure than decades of research, okay? So, and this is validated. So when you're playing this game, just know that it's actually following real fundamental laws of biochemistry and physics, which I think is pretty cool, okay? So there you go. That's folded. Please make sure you have it done before the final exam, just because I don't want you worrying about it after that. And I think it'll be a pretty good study tool too. But anyways, go to these websites right here, make an account and join our group so I can track your progress. Okay, that's very important though. Please be sure to join our group. If you don't join our group, Villanova CHE, I can't see your account. I can't see that you've completed the tutorials, okay? So anyways, that's folded. Now let's talk about what you're going to be doing in lab this week. So we just talked about all the levels of protein structure, and we ended with quaternary structure. So the assembly of different subunits into dimers, tetramers, etc. Okay, that's exactly what you're going to look at in lab this week. We're going to take this enzyme, beta-galactosidase, which normally forms a tetramer, and we're going to look at it with a technique called dynamic light scattering to measure the size of that tetramer, which you should find to be about 20 nanometers. Okay, so DLS is a really powerful technique where you pretty much just use a laser. You shine it at the sample, and then you're able to collect enough data from how the laser bounces off those particles to estimate its size. So you'll actually see that. You'll see data that tells you this enzyme is 20 nanometers in diameter. But we're going to take it a step further. We're also going to dimerize the protein, so dissociate it from a tetramer into a dimer at alkaline pH. So at pH 10, we start to see things that change in the protein. The conformation certainly might change, but if you get much higher than pH 10, you might start protonating the aspartate and the glutamate, which would then disrupt some of the ionic bonds between the subunits, right? So that would break those bonds, and now you get this dissociation into a dimer that's closer to about 10 nanometers instead, okay? So that's what you'll be doing in lab this week, is actually looking at and studying that phenomenon of quaternary structure in beta-galactosidase. Now, I should mention here that we've got a minor change of plans. So I'd previously said that we were going to study alpha galactosidase from human, but that proved to be too difficult, too expensive to obtain. So we're just going to resort to beta galactosidase. It's pretty much the same type of enzyme. It undergoes the same type of reaction. So it works for all the experiments I had planned, but it is dirt cheap next to human alpha gal. So yeah, that's the reality of the situation. Sorry for the confusion, but that's all you need to know. And by the way, everything that we're going to do for beta gal, we just as easily could have done with alpha galactosidase as well. Okay, so that's going to be the plan. Is when you meet with Sean, you're going to start with a tetramer. It's going to be 20 nanometers in diameter. You're going to increase the pH and then measure the size again. You're going to see the dissociation. Okay, and I think he also has planned for you too. You're going to look at the enzyme under different salt concentrations. So a high salt, 1 molar NaCl concentration where it forms this tetramer, and then a low salt concentration where it forms this aggregate. And so you'll see a very high diameter there, okay? Just to demonstrate the range of diameters that this instrument can detect, okay? Now, the reason all this is important is because a lot of times enzymes need to have a specific quaternary structure to function. So for example, beta-galactosidase might need to be a tetramer to undergo its reaction, to form its catalyst or active site that's required, right? So maybe the dimer is dysfunctional. Maybe it doesn't perform the reaction. Maybe the dimer is so small that if you were to inject it in the bloodstream, it would very quickly leave the bloodstream, okay? So protein size, the quaternary structure, can often be essential. And so that's why we need techniques like DLS to measure that size before we can put this drug on the market, for example. All right. And then, yeah, so since I'm spending all this time catching up from other lessons, I did just want to say real quick or explain how DLS works. So pretty much what you're going to do in lab is you're going to have your sample here containing your enzyme, and there's going to be a laser that shoots into that sample. And if there was nothing else in that cuvette, in that sample there, that laser would go straight through and to the other side. But if there are particles in that sample, the particles will scatter that light. Essentially, you'll have a particle here, a laser beam coming in, it hits that, and then bounces off it in a specific direction, okay, instead of going straight through. So there are detectors inside this instrument at angles other than the angle that goes straight through the sample, and they detect how much of the light is scattered. So that's why we call it light scattering. We want to detect what fraction of the light bounces off these particles. But that only gives us so much information. That just tells us something is present, right? If there's scattering, then there's some particles in solution, but it doesn't tell us the size of those particles. To get the size of the particles, we have to look at the data itself over time. Okay, so that's why we call this dynamic light scattering, is because we're not just taking a snapshot in time. That would be static light scattering. No, we're looking at the data over time. That makes it DLS, okay, dynamic. And what you'll see here is it might not be very intuitive, but if you have a particle with a large hydrodynamic diameter, that's this DH here, okay, so if you have a large particle, you'll see that the intensity of this scattered light is detected at a much higher magnitude. You see those peaks are going much higher and lower than if I have a smaller particle. You see how the magnitude over here is much smaller? So the larger particles scatter more light. Also, the other thing that you'll see is that over time, smaller particles tend to move around a lot more, okay? When you have that phenomenon, when the particles are moving faster, then you tend to see fluctuations in the scattering of light that happen at a higher frequency, okay? So it's really hard to see, but can you see how the distance between these peaks is wider, whereas the distance between these peaks is much narrower? That's because this small particle here moves back and forth really fast, whereas the large particle moves slower. So if it's scattering a bunch of light at one point, it's going to continue to scatter that light for a longer period of time than if we have the smaller particle over here that's bouncing around. You know, it might have quite a long period of time where it's, or it might have some periods where it's out of the path of the laser, but then back into it. Okay, that's probably the best way to describe it, right? When the intensity is low here, that particle is not scattering the light. When it's high, it is. So it might bounce into the beam and then out and out and out. And that's what we're capturing with the DLS. But even with that, even if you're able to understand that jargon, how do we get to size? Well, there's no easy way for me to explain it. The software does most of that, and we honestly don't have time for me to dig into the details, but that's what happens. The computer is able to look at these profiles, look at the magnitude, the frequency of those peaks, and give you estimates of the size. So a profile looks something like this. It will estimate as having a size of, let's see, what is that, about 8 nanometers, whereas a profile like this, it will estimate to be around 1, 2, 3, 4, we'll call that 4.5 nanometers, okay? That's the data that you'll actually see on the DLS, all right? I don't know if Sean will show you the other real-time, the dynamic data, but that's what it allows you to do. And so it's perfect for this experiment that we're trying to plan, right? We want to detect the difference between a large tetramer and a smaller dimer. So the instrument will do all this for us. It will give you a plot that looks something like the one down there, and you'll be able to actually measure the size of the tetramer at pH 7. But then when it dissociates at pH 10, you'll be able to see that the size is about half of the tetramer, which should make sense, right? Because it's half of the particle. All right. So yeah, please do let me or Sean know if you have any questions on that, but that's what we have in schedule for lab this week. Okay, so that closes out Monday's lesson. Now I just want to switch gears pretty abruptly and go back to the lesson from Monday of last week, right before our exam. So that's when we were talking about protein detection. And we got pretty much all the way through PAGE, and we started talking a little bit about LCMS. But there were some very important slides at the end that I need to go over with you, okay? I cannot have you go through this class and not talk about Western blots and ELISAs. It's fundamental to what we do. You have to know how these things work, okay? So let's go back and now talk about Western blots and ELISAs for protein detection, okay? Whoops. So we had talked about PAGE shells, right? And a PAGE shell is nice because you can put samples onto the gel and then you can run it through an electric field and it will separate the proteins based on molecular weight. If you run a 2D gel, it separates them based on PI as well, okay? But even so, even if you do 2 dimensions, there still can be some pretty big question marks, right? You stain that gel and you can see there's proteins present, but you don't know what those proteins are. You could certainly find that out, right? We talked about using LCMS for that. You can cut out one of those bands or one of those spots from a gel, and then you can do a tryptic digest. You can do the mass spectrometry and get a pretty good idea of its identity, but there's a lot of work involved in that. It doesn't work perfectly sometimes, and it requires a mass spectrometer that's going to be hundreds of thousands, if not maybe $1 million in total cost. So that might be prohibitive for some labs. So wouldn't it be nice if we had an alternative to that? If we had an alternative way to look at a gel and decide, is this band right here my target protein? Okay, I want to be able to do that at low cost without having to have a mass spectrometer. And that's where Western blots come in, okay? So what Western blots do here is we take everything that's on a gel, okay? So let's say we have a gel here, and we're gonna look at that gel from the side. So your bands are in this gel. And then we're gonna sandwich it together with a nitrocellulose membrane, okay? And then we're gonna apply an electric field to draw those proteins out of the gel and then onto the surface of the nitrocellulose membrane, okay? When you do that, then you can remove the membrane. Now your proteins are on the surface of the membrane, okay? And you can label them with an antibody. And what the antibody will do is it will specifically bind to us one protein, right? So if we were looking for our target protein, we would have an antibody that we know binds to our target and nothing else. And if that antibody is labeled, then now the antibody we can visualize on that sheet, on that nitrocellulose membrane, and we can definitively tell that's our protein, right? Our protein is present in that sample. So that's a process that only takes a few hours, and really all you need for it is the electrophoresis system that you already have, that you already use to run the gel, and a rocker and an antibody. That's pretty much it. So it's pretty inexpensive and easy to do, okay? So that's a Western blot. You take the proteins off a gel, transfer them to a nitrocellulose sheet, and then label that with an antibody. At least that's the overview of it. There's a few other details that I'd like to share with you here. So in this example, we've run a one-dimensional page gel, and some of these lanes have our protein. Now we can't see it. I'm just making it green there to illustrate the point. But we want to use a Western blot to confirm that that green band right there is our protein. Okay. Like I said before, we're going to take that gel, we're going to sandwich it with a nitrocellulose membrane, apply the electric field, the proteins get transported over to the membrane. At that point, we can't see them. They haven't been stained with Kumasi or any other type of stain, but they are there. And that's what these dashed boxes are indicating, okay? Now, what's very important is at this step, we have a nitrocellulose membrane that is covered in some areas with protein, right? The proteins that came off the gel are now on the surface of that nitrocellulose. But there's a lot of other unused area on the nitrocellulose that's just blank. And that is potentially a problem because nitrocellulose, the reason we're using it here is because it binds to protein with a high affinity. So that's great for when I'm transferring the peptides over. I want them to stick to the nitrocellulose. But remember, the subsequent step here is going to be to add a labeled antibody to this nitrocellulose membrane. So if I have blank areas of nitrocellulose, guess where that antibody would end up? I'd probably have antibody everywhere else except for on these proteins. Okay, I can't have that. I need to localize antibody binding just to my target protein. So the way I'm going to do that is I'm going to take my nitrocellulose membrane that has all these protein bands on it, and I'm going to cover it with milk. There's a lot of proteins in milk. So pretty much we're just non-specifically blocking the rest of the unoccupied surface on that nitrocellulose. So all this gray area is milk protein. We don't have to be fancy about it. We used to keep dehydrated milk from the grocery store in our lab when we were for these Western blots. But it donated the protein, right, that kept the antibody from binding to the other parts of the nitrocellulose. Okay, so we're going to block it with milk. And then we're going to apply not just one antibody, but two antibodies here. Bear with me. The first antibody is going to be one that's specific for your target protein, okay? So this is the target, that little green ball on the surface of the nitrocellulose, okay? So same thing we're talking about in the page shell up there. When we apply what we call the primary or the first antibody, it binds to that target, okay? So now we've got our complex, and that antibody is only bound to your target protein, nowhere else on the nitrocellulose. We wash that off, make sure we get rid of any excess antibody. We don't want any of that floating around anymore. And then we apply a secondary antibody, and the secondary antibody binds to the primary antibody, okay? Now you might wonder, whoa, antibodies are pretty expensive. Why are we sandwiching them together like this? Why is that necessary? It seems like it's wasteful. But what's going to happen here with the secondary antibody is that we typically have some other type of functionality on that secondary antibody. So for example, it might be linked to another protein, something like horseradish peroxidase. That's an enzyme that can take a colorless substrate and turn it into a colorful dye. Okay, so if my secondary antibody has HRP on it, then I wash everything off, and then I add this colorless substrate, and then only where my antibodies are bound to my target protein will I see the formation of that dye on the surface of the nitrocellulose. Okay, so in this case, the primary antibody is serving the function of binding specifically to the target, and then the secondary antibody brings in some sort of marker. an enzyme, like is shown in this case. It could also be, sometimes you'll see fluorescent antibodies. So then you don't even need a substrate. You could just have a fluorescent antibody and visualize it under whatever kind of light excites that fluorophore, and you would see it light up wherever the antibody is. Back in the day, we used to have radioactive antibodies that we would use for this purpose. Believe it or not, I've used those in lab. And we used to take radioactive film and just set it on top of these things, and overnight, the radiation would expose that film and we would be able to see where the protein was. So there's a lot of different ways to do this. But that's the basic approach. The primary antibody binds the target, secondary antibody binds the primary antibody and gives you some sort of signal. Now, a very valid question at this point might be, why don't we just put HRP on the primary antibody? Why don't we just put the fluorophore there? Why not? Why not take out that additional step there with the secondary antibody? Well, the answer is you certainly could. Be my guest. You could put anything you want on that primary antibody and skip that secondary antibody step. However, that would mean that you would have to have primary antibodies for every different protein you'd ever want to detect conjugated to HRP or to a fluorophore. There is some chemistry there that you certainly can do, but it's somewhat complicated, it's arduous, and so people tend not to do that. Instead, what they do is they just acknowledge that there's going to be antibodies for virtually any protein. That's going to be what we're going to use as a primary antibody. And then all you need for your secondary antibody is a secondary antibody that generically binds to any other antibody. You see how that simplifies this now? Then a company, something like Thermo Fisher or Abcam, for example, can make huge batches of a secondary antibody that are conjugated to something like HRP or a fluorophore, okay? And then they can sell those to use in combination with these very more specific primary antibodies, okay? So overall, it's just an efficiency sort of thing. Instead of taking the time to label every single primary antibody, I just label a bunch of generic secondary antibody, and then I can use that with any primary. It's going to be more expensive, but it's going to save you time. And so that's the procedure that we typically use. Okay, so that's Western blot. That's how we take a look at a page gel and its myriad of different bands, and we use a pair of antibodies to specifically determine where our target protein is. Okay. And this is what a typical result might look like. So if you remember our case study from a week and a half ago, what we were trying to do all this for was to try to detect proteins that were linked to cancer. And so you could, for example, have patient samples from a variety of tissues. And you might want to know, is protein X present in all those tissues and in those samples? And if it is, that patient might have that type of cancer. So in this Western blot right here, you might have patients with prostate, breast, and pancreatic tissue samples, right? And the dark band here would be from that HRP. So that's telling you that, oops, that protein is present because it's being bound by the primary and the secondary antibody, giving you that HRP signal. But then you look at this sample over here from lung, and you don't have that protein present. So good, that patient doesn't have cancer. Okay, and so this is something that you could quickly, easily do to determine if that protein is present and give that patient a diagnosis. But this also, I say quickly, but it still would take several hours to run the page gel, right? You guys have done that. To make that gel, to run it took us 3 hours of an afternoon. Then you got to go through all those other steps, transferring to the membrane, blocking it with the milk, applying the series of antibodies. A Western blot is really an 8 to 12 hour process. Some people run it overnight too, certain steps of it. So there's a time consideration there still. It's inexpensive, but it takes a lot of time. So wouldn't it be nice if we had an alternative method. Maybe one that didn't necessarily rely on page shells. We could cut that time out, right? And, oh, let me just skip all this. That's where ELISA comes in. And ELISA pretty much does the same thing for us as a Western blot. It tells us if a specific protein is present in a sample, but it does it in a completely different way. We're still going to use antibodies here, but the order with which we add everything is going to be flip-flopped a little bit, okay? In ELISA, what we do is we take an antibody that we know binds to protein X, whatever we're looking for, and we immobilize that onto a surface. So this could be the plastic bottom of a 96 well plate, for example, okay? We're going to immobilize that antibody first. Then we're going to take the same kind of sample that you might put on a page gel, and we're going to put that into that well on a 96 well plate. If our protein, the target protein, is present, it will quickly be bound by the antibody. Okay, actually, here we go. We'll get something that looks like this. Okay, so that's nice, but it's not very exciting because we don't really have a signal yet, right? We don't have a way to detect that interaction. So this is where the next step comes in, which is a secondary antibody with a marker. Now, be careful here. In the case of the Western, we had the antigen that was immobilized first, then we had our primary antibody, then we had our secondary antibody, okay? Notice the difference here. Instead of the secondary antibody binding the primary, in an ELISA, both the primary and the secondary antibodies bind to that target protein. Okay, and they shouldn't bind to the same spot. You're going to need two antibodies that bind to opposite sides or just different sides of the protein. Okay, if that occurs, if you have those antibodies, then you'll get a complex that looks something like this. Okay, and if I label that secondary antibody with some sort of marker, which could be colorimetric, fluorescent, or even enzymatic, then now I can detect if that protein is present, right? Think about this. If and only if that antigen, that target protein is present, will the secondary antibody be immobilized as well, okay? So if I add that secondary antibody to my well and I wash it thoroughly, right? I add it, I let it incubate for a little while, then I remove that liquid, I rinse it with something like phosphate buffer saline a few times, okay? If and only if the target protein is present, will that secondary antibody stay in the well? If that is not present, it'll get washed away with everything else, okay? So that's how this concept of an ELISA works. You have to form this complex of primary antibody, antigen, secondary antibody, and then you have some sort of signal that tells you everything's there, all right? That is a method that I can do very quickly and very inexpensively. Yes, antibodies are relatively expensive, but if I set this up just right, I can use a sparingly small amount of antibodies, okay? Just a few antibodies with the right type of marker. If this marker can amplify this signal, if it's a very bright fluorophore, for example, then I don't need much of it, all right? And notice I've completely cut out the page gel, the nitrocellulose membrane here. All I had to do was add primary antibody, add the sample, add secondary antibody, and we see what we've got. I've skipped some wash steps in there, but it's pretty simple, right? So this is exactly how COVID tests, and well, the example I have here is a pregnancy test, work. If you've taken a rapid test, you've done this, but the canned example I have is pregnancy test, so let's just go along with that. What happens here with this type of ELISA is you're trying to detect a marker. In the case of pregnancy, it's going to be a hormone, okay? So pregnant women start to produce a hormone that's not there before they're pregnant. So these primary antibodies here in the middle, they specifically bind to that hormone, okay? So if I take a sample and I put that onto the pregnancy test, that hormone is going to bind to those primary antibodies. Now, at the same time, the application of that sample is going to weaken the bonds that are holding these antibodies onto the surface of that test, of that strip, okay? So these bonds are going to be broken, thereby releasing that secondary antibody shown in red. So the secondary antibodies are going to flow over the surface of this test strip, and if that hormone is present, they'll bind to it there, and so I've formed my structure, okay? And in this case, those secondary antibodies that I have colored red, they would be dyed somehow. They would be conjugated to something that you could see with the naked eye. So you don't need to add a substrate, wait for an enzymatic reaction. You don't need a fluorescent microscope or anything like that. You'd be able to detect that with your eye. Okay, that's why they're colored red. Now, that would indicate, so if you saw red on this region of the test strip, that would indicate that the hormone is present, right? And the patient is pregnant. Congratulations. However, we need to be careful here, especially with a question like this of, is the patient pregnant or not? There's always a chance that tests like this that are stored on the shelf for long periods of time, they might expire. These antibodies that are required for all of this might denature and then be dysfunctional. If that's the case, then you could have a woman that is pregnant, but since the antibodies aren't working, they would get a false negative result, right? These antibodies wouldn't bind here, so you wouldn't see them. Okay, so we want to prevent the possibility of a false negative, or at the very least, if we do have a false negative, we know something's wrong. Okay, so with one of these pregnancy tests, what you're going to do is you're not just going to have primary and secondary antibodies here, you're going to have a third zone with control antibodies. And what these control antibodies do is they're able to bind the secondary antibody. Okay, so if we have any excess antibody, secondary antibody, that will flow down here and it will bind to this control region. So you should always see in this region a red strip. Okay, what that red strip tells you is that the antibodies are present and functional. Okay, if you have a pregnancy test where you don't see a red strip in the control, that means it's an invalid test. It would give you a false negative or just no result at all. Okay, so if you have a result here in both spots, then congratulations, the test is valid and you're pregnant. Okay, so you want to have all of those, you want to have that control in place just to verify the test. Oh, and here we go. That's exactly what you see. Okay, so pregnant, we have bands at both spots. If you're not pregnant, you do see the band with the control, but you don't see the band in the actual test region there. Okay. And yeah, I think that's everything. So yeah, I just went over how an ELISA works for a pregnancy test, but we routinely run ELISAs to detect proteins in samples in 96 well formats as well. So it doesn't have to be a test strip. Everybody's familiar with pregnancy tests and at least COVID tests, right? And how the rapid tests work, but you can do it in either format, all right? But just be able to explain on the exam, for example, what the difference between a Western and ELISA is. You need to know what an ELISA is because you're going to do one at some point in your future career, probably, or at least understand how they work because you might be selling them. All right. So I think I'm actually going to stop here, save this video and upload it. And then I'm going to prepare another video that walks you through what to do with our data from lab last week. Okay, so everything you need to know for your lab report. All right, so look for that on Blackboard. There's going to be a second video after this one. It tells you how to analyze your data. All right, I'll see you there.